Okay, so now that we have understood how individual amino acids bond, let's talk about really how the protein is organized into a final structure that's going to have some sort of biochemical significance. So there's really four levels to protein structure. We think of the protein primary structure, we think of the protein secondary structure, tertiary structure, and the quaternary structure. Okay, so I'm going to write them out just so that I can muse about it with you, and then I'm going to show you a more formal diagram and a more formal video to help just bring this all together in a really true scientific way. But I wanted to leave this open-ended at first so that I could have a discussion with you about it because I really love this stuff. So primary structure really refers to just the sequence of the amino acids. Okay, so we have amino acid here, amino acid here, amino acid here, amino acid here. So let's say we have a 5. Let's get crazy. Let's do a 10 peptide amino acid. Why not? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so these are all amino acids. We know that we have the N terminus over here, and we have the C terminus over here, and we know that there is that peptide bond, that amide bond. So the primary structure really just refers to the sequence of amino acids coded by the genes. Okay, so we have the codons. What is the ultimate sequence of the amino acids? Because it's this sequence that is ultimately going to determine the function. I mean, if we only have 4 base pairs coded by triplets of codons leading to one amino acid, then the only thing we can vary is the order that those things show up in the genome. So the sequence of proteins really is everything. So the sequence, the order of these proteins is what matters. Okay, like if we had a methionine here and a tyrosine here and then a aspartamine here. So the primary structure really refers to the sequence. Okay. Now, the secondary structure refers to how, once you have that sequence, how that region folds on itself. Okay, so we have that primary structure, and then we have the secondary structure, which is the shape that that sequence itself forms, that adjacent amino acids form. And there's really two ways that the sequence can create that initial three-dimensional shape. There's something called the alpha helix, and then there's something called the beta sheet. Sometimes you see beta pleat sheets, right? So an alpha helix is when that structure forces the amino acids to form in somewhat of a helix like this. So these are, individual amino acids, and this is actually stabilized by things called hydrogen bonds. Okay, and that's you have these structured hydrogen bonds between the different side chains, which stabilize it. Okay, and another way that secondary structures can form is they can form according to these things called beta pleat sheets. And in a beta sheet, it's kind of tricky to draw. Let me peek down here and see how they're doing it. Yeah, they're doing it like that. I don't really like that drawing. I like just to think of the beta sheet as the protein sort of forming like this, where you have these little side chains here stabilized by hydrogen bonds that way. Hydrogen bonds are what are called intermolecular forces. If you had me for chemistry, you understand that. If not, let's not worry about it. They're not bonds. They're really just forces of interaction. And we're going to be playing with a game called Fold It next week that's going to allow you to play with that. So in terms of structure, we have this primary structure, which is the sequence of amino acids. We have the secondary structure, which is the three-dimensional shape that happens once that sequence is established. We have alpha helix and beta sheets. And then we have the tertiary structure, which is how that ultimate sequence and those three-dimensional shapes fold on themselves. So let's say we have a long protein made up of 200 amino acids. We might have one region where it forms an alpha helix, and then another region that might conform to a beta sheet, and then another region that might conform to an alpha helix. And this is going to lead to a larger folded protein, right? So based on those positions, we might have one region of the protein, which is an alpha helix, and then it might collapse in on itself to create these beta sheets. And then we might have another region that's an alpha helix leading to a beta sheet that might go like this, and then another alpha helix out here. Okay, so the tertiary structure is how the three-dimensional structure of the sequence folds in on itself, and this structure here is very important. I mean, this region right here might be a binding site for a protein. And now the quaternary structure is what happens when multiple folded regions of a protein come together. Okay, so you might have a gene, and that gene might code for a protein, but that protein might have two different tertiary structures to it. So there might be two regions. So perhaps one region of the protein is an alpha helix with a beta sheet, okay? And then the other protein maybe is a smaller alpha helix with a beta sheet. And you can think of this as one tertiary structure, and you can think of this as another tertiary structure, okay, that's connected. And then this entire thing here is the protein itself, okay? So we see this with hemoglobin. There is one gene coding for hemoglobin, but hemoglobin has 4 different subunits, 4 different tertiary structures, which then come together to make the quaternary structure. So you can imagine a gene with various start and stop codons interspliced throughout the entire gene. Okay, so in review, the sequence of the amino acid, just the sequence, not thinking about the three-dimensional structure, just the genome, is called the primary structure. How those individual regions fold in on themselves creates that tertiary structure, and then how those, creates that secondary structure, and then how those secondary structures fold on themselves creates the tertiary structure, and then if there's more than one tertiary structure, we then come together to form the overall quaternary structure. Because this is probably a cruddy video by me, I went ahead here and put a screenshot from the internets that I think is a little bit better. This is indicating that, like I said, the sequence, okay, of the amino acids is the primary structure. The secondary structure is how this hydrogen bonding creates these repeating patterns, okay, of three-dimensional structures. Okay, we have those hydrogen bonds sort of holding these backbones together. They're drawing their beta pleat sheets a little different. They're calling the beta sheet a pleated sheet. This is the beta sheet, same thing. Okay. The tertiary structure, they're showing how there's that three-dimensional folding due to the interactions of these side chains. So it's showing in here regions of alpha helix and then regions of beta pleat sheet. I like my drawing a little bit better actually there. And I could do this here to help you. Okay. And then the tertiary or the quaternary structure is showing how proteins consisting of more than one amino acid side chain. So what we have here is one tertiary structure right here and the other tertiary structure right here. So in summary, these levels refer to 1, the primary structure, the sequence. 2, the secondary structure, that backbone supported by hydrogen bonding that creates our initial three-dimensional look at it. And then the tertiary structure, how that backbone folds in on itself to create a shape, which really is ultimately going to lead to the function of the protein. And then the quaternary structure, how those individual levels come together. Okay?